Good morning. We're still talking about genetic diseases, specifically those caused by errors in enzymes that affect metabolic pathways. And we're going to use PKU as our example to talk in a little bit more detail about the kinds of alleles that occur in the human population and the effect that those particular mutations have on the functions of this particular protein. So reminding you about PKU, this is an example where there's a mutation in the gene and it affects the catalytic function of the enzyme that's eventually produced from the mRNA that's, of course, translated to form the protein. Okay, so this is a real example. It's phenylalanine hydroxylase. The gene is located on chromosome 12. And the mutations are cause of a very severe genetic disease called phenylketonuria, PKU. And this is one that we like because there's a successful treatment for it, and these therapeutic interventions largely prevent disease progression. Reminder, the enzyme phenylalanine hydroxylase simply takes the amino acid phenylalanine and converts it to tyrosine as part of our metabolism of dietary proteins. So in summary, dietary protein is a source of most of the amino acids that we have in our bodies, including phenylalanine and tyrosine. In the case of PKU, what happens is the phenylalanine is largely converted to tyrosine as a way of metabolically disposing of the excess. Tyrosine itself can lead to the production of other important substances as well as proteins in the body. In the absence of the phenylalanine hydroxylase enzyme, the phenylalanine is not converted to tyrosine, and the excess cannot be disposed of or metabolized to other important compounds. Instead, you get accumulation of phenylalanine and phenylpyruvic acid and other metabolites in the body, and this has very serious effects on the developing nervous system. Okay, so this is typical of this sort of genetic disease, that as a result of the deficiency in the enzyme, the pathway stops at that point. You accumulate phenylalanine, and you also convert some of it to other toxic metabolites which are not normally present at high concentrations. Reminder that high protein foods are important to avoid if the person has PKU, because you have to control the amount of phenylalanine that's coming in through their diet. So essentially no natural sources of proteins are permitted, only low phenylalanine foods such as fruits and vegetables. You do, of course, need phenylalanine for synthesis of your proteins, but that is provided by a special formula which is consumed several times a day. So one can buy these phenol-free formulas and make milkshakes and things out of them. And that's not so great for a kid, or try to convince your kid that they can't eat anything with protein in it. Things have gotten a little bit better because there are actually some special PKU foods that are made with low amounts of phenylalanine that are a lot more palatable than just amino acid milkshakes. It's not a very rare disease by any means. The carrier frequency among Irish is 1 in 33, and also in other Northern Europeans. 1 in 12,000 births in the United States is a PKU baby. Okay, so it's inherited as autosomal recessive, and we're familiar with the big A, big A, big A, little a, little a, little a nomenclature. But what we want to talk about today more is how many different ways there are of being little a, little a. If the little a allele is one that does not produce a functional enzyme or produces a less than normally active one, then two copies of that particular allele will have the disease. But there's different ways to be little a, as we'll see, because there's a lot of different alleles out there in the population. Okay, many different alleles means many different effects. Some are very mild, others are very serious. Classic PKU is one where very little or no active enzyme is present. There's variant PKU, where there's some amount of enzymatic activity, but not enough to be normal. Then there are cases, depending on what mutations you have, of non-PKU hyperphenylalanemia, where you have elevated phenylalanine, but not enough to cause a problem. There are also, with all the sequencing that's going on, a lot of variants of unknown significance, that they change parts of amino acids in the protein, but there's no easy way to attribute particular defects to those mutations unless you actually look carefully at the protein. And there's just too many of them. Okay, in reference to the gene, it's pretty big. It's 90 kilobases roughly, 90 kilobases. The primary transcript's about 85,000, but the mature mRNA is only 2,800 base pairs. So most of the primary transcript is removed during splicing. The introns are huge compared to the exons. And in the end, the protein has 452 amino acids. Okay, you don't have to see what you can see on this map. Each little dot represents a different allele that's been discovered. On the bottom, you see the more or less scale-drawn size of the primary transcript, and the little teeny little strips of black in the middle of those are the exons. So the introns are humongous in this protein and very plentiful. Okay, so more than 500 different variants are known. Some of them are harmless polymorphisms, and others cause very severe disease if homozygous or coupled with other harmful alleles. So I want to go through some of these and really just get through the nomenclature of how we talk about these mutations. Okay, so there's many ways to be little a. Some people are what are called compound heterozygotes, which is to say they've got two deficient alleles, one from mom and one from dad, but the alleles themselves are different from each other. They contain different mutations that have different effects on the protein, but nonetheless, neither one is active, and so the person will have the disease. Okay, reminder, missense mutations change the nucleotide sequence to produce an amino acid change. Silent mutations change one codon to another that encodes the same amino acid. And then you have nonsense mutations, of course, where a sense codon is changed to a stop or nonsense codon. Okay, so the most common allele, the classic severe PKU that's most well studied, is called the R408W allele. So what that means is R means the amino acid in the original form was an arginine. The 408 means it's amino acid position 408 in the protein, and the W means it now reads as tryptophan. So in this case, the codon change goes from a CGG to a TGG, which is a missense mutation, of course. Okay, so here's what the exon looks like. This is an exon 12. The CGG codon, that's the original, has been underlined, and that in the R408W allele is now reading TGG, or UGG in the mRNA, and that encodes tryptophan. And that's a very severe allele, the cause of classic PKU, the most common allele. Okay, so here's a picture of the protein. Amino acid 408 is bold-faced and underlined. It's close to the C-terminus of the protein, and so the R in that bad allele is now reading as a W or tryptophan. Another severe allele is the R111X mutation. The X is another way of saying stop codon, so it's a nonsense mutation. So in this particular mutant, the original codon is CGA, encoding arginine, and the mutation is reading as TGA, which is a stop codon, which is going to, of course, terminate the synthesis of the protein at that codon. Right, so that one's in the second line, the arginine here in boldface and underlined, is the one that will be changed on that particular allele. Okay, now the consequences of that are, of course, pretty serious because translation will not reinitiate on the mRNA once it encounters that stop codon that's substituting for the arginine codon. Okay, so the question, how many amino acids will the protein contain that's produced from the R111X allele? 110, 111, 112, 452. Okay, so it's 110, because what would have been amino acid 111 is now a stop codon, so the protein itself has 110 amino acids. Okay. When a person is homozygous for the R111X mutation, they will have classic PKU, mild PKU, non-PKU hyperphenylalanemia. Right, A. Classic PKU, no functional protein can be produced, so that will be a very severe form of this disease. Here's another picture. Okay. Okay. I94 delta. Okay, this is a very mild allele. There's an isoleucine at position 94, and it's missing. So the protein is normal in every other way. And it's a lucky deletion because the 3 nucleotides removal means you're not throwing the translation out of frame, so the rest of the mRNA can still be read in the correct reading frame. So that's not a problem, not a severe problem at least, with this particular protein. Okay, and that's there in the first line with the underlined isoleucine there is simply missing. Okay, you can also have frame shifts and other larger mutations, etc. Okay, this particular splicing mutation is particularly awful. Changing nucleotides at the GUAG intron borders will prevent splicing from occurring correctly, and if you fail to remove an intron correctly, you will prevent good production of the protein. Okay, here's an example of a splicing mutation. In this case, intron 12 has an AU instead of GU at the intended 5' splice donor site. Okay, this is a sort of odd representation where the normal version of splicing is shown at the top of the picture, where you have your GU, AG, as you should have, in the intron between exons 12 and 13. In the mutant form, that GU, instead of reading GU, reads AU or AT at the beginning of that intron. Now we know that the GU-AG rule guarantees splicing to occur correctly at those intron exon borders. So the effect of the mutation is not really what you would predict, I don't think, because what happens is, as diagrammed, exon 12 is skipped entirely. So the splicing goes correctly at exon 11, but instead of going to exon 12, it skips it completely and goes directly to exon 13. So that's going to be a severe mutation because you're missing that entire exon. There are also mutations that affect transcription. It's not the usual kind of mutation that you think of in these cases, but it certainly happens. That you could have major effects on whether the gene is transcribed at all, depending on what's happened to your upstream region. Now, some changes have no effects, but some of them can be very devastating, as in this example. So there is an allele out there where 3.7 kb of DNA upstream of the transcribed region is completely missing. So there's basically no promoter, no enhancer, no nothing, and so there's very little transcription from the gene. The coding region is perfectly fine. If you sequence that, you don't have any mutations in it. But nonetheless, if you can't make your RNA, you're certainly not going to make your protein, and this leads to a severe deficiency in this particular protein, obviously. Okay, so here's an example to remind you of compound heterozygotes. A person has PKU. Therefore, they have 2 little a alleles, but these can be different. So in this example, mom, the little a defective allele, is the R408W classic severe PKU. And from dad, the little a is an R111X mutation, which is to say a stop codon at position that would have been amino acid 111. So diagramming those proteins, mom's got a normal length protein, but she's got a mutation at that one position in the protein. Dad has a shortened protein. He's only got 110 amino acids, and neither protein is functional. So this person is going to be little a, and have a disease, even though the mutations from the 2 parents are different from one another. Okay, I just probably said the answer to this, but a person got the R111X allele from dad and an R408W from his mom. Phenotypically, he is normal, has mild PKU, has classic PKU. Yes, he has classic PKU. Both alleles produce very severe disease. A person got the R111X allele from dad and a wild type from his mom. Phenotypically, he is normal, has mild PKU, or has classic PKU. Well, he is normal. He will have less of the functional enzyme than a normal wild type person would have, but it's enough to avoid any phenotypes or any disease severity. Okay, so there are some newer treatments to this beyond the really grimly difficult protein-free diet, which don't really attack the problem directly, but get at it obliquely in kind of clever ways. For one thing, this drug, Kuvan, which is recent, is good for what's called BH4-responsive PKU. And that BH4-responsive PKU generally results from forms of the disease which have some enzyme activity already. If the enzyme is completely absent, it's not going to help you. But if there's some activity, it can be boosted by having extra amounts of BH4, which I'll show you in a second. The other interesting new treatment is an enzyme substitution therapy, which is a drug that degrades the extra phenylalanine. It's not phenylalanine hydroxylase. It's a completely different enzyme. So we have to look at the structure of this protein to understand something about why you would have BH4 responsive. On the left, you have the monomer form, which has a catalytic domain, a regulatory domain, and a tetramerization domain. So the final enzyme is actually formed from 4 copies of this monomer that form together into this tetrameric complex, which is the active enzyme. So this is kind of a finicky enzyme, and I think that's part of the problems that people have been having trying to produce it or making therapy or whatever for it. Okay, so we've got the regulatory region, the catalytic region, and the tetramerization. The folding of this protein is apparently quite difficult. So the part of the protein that binds the cofactor, BH4, is in the catalytic region because it's a cofactor in the reaction. So it turns out that many mutations cause amino acid changes that lead to structural changes that cause the protein to be degraded rather than directly influence the catalytic activity. Misfolding causes a lot of problems with this particular. A lot of alleles cause misfolding. So the problem is nothing directly to do with the catalytic reaction, just that the protein is not folded correctly. Okay, so sort of representing this in a sort of silly diagram, if you think about the protein as it's coming off the ribosome, it's in the process of getting made, that it has to start folding at that point. And you can have a correct folding intermediate, which is what you're supposed to have, so your monomer then will associate with another copy of itself to form a dimer, which assembles with 2 more copies to form a tetramer, which finishes folding to form an active tetramer. Now, things can go wrong with this protein in particular for many reasons, where in the initial stages, you might get a little bit of misfolding, which kind of takes the rest of the process and drives it into the wrong direction, where you have the monomer then associates with other misfolded monomers, and then eventually aggregates to a completely inactive form. Okay, so BH4 is an example of a pharmacological chaperone. It's a naturally it's tetrahydrobiopterin, and it's a cofactor for the reaction. That's a necessary cofactor for the reaction. And of course, as a cofactor for the reaction, it does bind to the phenylalanine hydroxylase protein. But in the process of binding to it, it stabilizes the protein and is able to avoid some of the extra misfolding that can occur with some of the alleles that are produced. So you're stabilizing or helping to fold, really, which is what a chaperone does. You're assisting in the folding of these proteins, leading to more accumulation of an active form rather than a misaggregated form, which are not active. So the idea is the addition is able to boost the activity of a weak enzyme, therefore reducing phenylalanine accumulation. So it is believed that it binds to the protein soon after synthesis, and doesn't just stabilize the fully synthesized protein, but actually assists in the correct folding of it in the first place. Okay, so this is, of course, especially helpful for mild forms of PKU, but people that are taking this drug, which is really a vitamin, are still supposed to stay on a low phenylalanine diet. But it is still very helpful for people whose form of PKU is responsive to this vitamin. Okay, the other interesting new treatment is a phenylalanine lowering enzyme. So this enzyme is phenylalanine ammonia lyase. This is not a human enzyme. It's found in yeast, some plants, and some bacteria. But it turns out to be helpful for people with PKU because if it's injected into their bloodstream, they're able to the enzyme can degrade some of the extra PKU that's around. So the idea is that the phenylalanine is degraded into harmless compounds by this enzyme that are then excreted from the body. And it does seem to work well, except for some allergic reactions that some people have had to it. So envisioning this situation, you have phenylalanine. The phenylalanine ammonia lyase converts it to tranexamic acid, which is a harmless metabolite, which is then excreted. And so the phenylalanine doesn't accumulate nearly as much if you've got this other enzyme to drive it away into harmless compounds. Okay, so where that sits in this pathway, if you have phenylalanine at the top, it's got different possible fates. The phenylalanine hydroxylase step naturally converts it to tyrosine and then to other compounds and eventually gets excreted. So that's the normal pathway, and that's what's blocked by the phenylalanine hydroxylase mutation. So in the absence of that, we're going to get accumulation of phenylalanine, with, of course, accumulation also of the phenylpyruvic acid, etc. So the point of the tranexamic acid, or ammonia lyase pathway, of course, is to take some of this extra phenylalanine and drive it off to the side into forming this harmless tranexamate, which will then, of course, be excreted and lead to reduction in the phenylalanine levels. So it's not a treatment, it's a treatment, not a cure, but it does seem to help some people. Okay, just to show you that this works, this is data from a phase 2 clinical trial of this drug, which is called Phe-lowering. Okay, and what's being measured is phenylalanine levels in the bloodstream of people that are being treated with this drug. So at 0 time, and what we're looking at is this orange line, where that's phenylalanine levels, which starts off very high, it's over 1200, which is way higher than the normal, it hoped for 360 micrograms per liter. And then upon treatment or addition of this Phe-lowering at 0 time, which the Phe-lowering, of course, is increasing over time because you're getting weekly doses, the phenylalanine level in the blood of the people decreases dramatically to the point where it's just about what you want for a normal person. And this is without really controlling your diet with protein. So this is an effective treatment, at least for some people. They should probably try harder to control their protein intake, but it obviously works reasonably well to control phenylalanine levels. Okay. So the Phe-lowering treatment, and they got closer to acceptable, and this has been approved by the FDA and is now on the market. All right, so other things that you would really like to do is give the person the enzyme, their human enzyme that they are missing. And this is such a difficult enzyme and so difficult to work with that this really hasn't been successful yet. Some enzymes are easier to deal with than others. And the other, of course, is to give the person a wild-type version of the gene so they can make their own phenylalanine hydroxylase. But this hasn't worked yet either. But people are still working on these things, and there's hope for the future.